Good morning to all of you. Today I will start the meeting. Uh, Sorry. Today I will just guide on to all of you on the gametogenesis. Okay. Uh, Gametogenesis is what the revision आहे ही just actually. Okay. The gametogenesis is what in body two type of cells are present. One is the germ and the other is somatic cells. And from that germ cells there is a formation of two gametes. There is a formation of two gametes which are there, male gamete and the female gamete is there. And in this slide, we review on the sex determination. Then just introductory part of the sex determination, which factors are responsible for it. That is a FAME3 genes are responsible for it. X and Y chromosomes are important. Like this way, there is a role of this into
आज
the sex
कोण आहे गे?
determination is there.
मी नाही येणार यार तिकडे उठून.
कोण आहे ते? म्यूट करा ते. म्यूट करा. अनम्यूट करा त्यांना. ठीक आहे. Difference between the spermatogenesis and oogenesis is there. The slight difference is there. Then external fertilization there. This is a general feature of the external fertilization. This is the spermatogenesis is there. काल हे मी तुम्हाला सांगितलं होतं. And the structure of egg of the CR gene हे पण झालेलं आहे. In this we are looking after about the chemo attraction. First step into the external fertilization. Then second step is the acrosomal reaction. Third step is the binding of the sperm to the vitelline membrane, then binding to vitelline envelope, then fusion of acrosomal process membrane and the egg membrane, and then afterwards there is a binding of the sperm membrane to the egg membrane is there, and then fusion of the female pronuclei to the male pronuclei. It happens, and this is the one question in every exam. In yesterday's lecture also I will told about to all of you that this is the very common question in every exam. It can be asked on the external fertilization. Today we will discuss about these EBR1 receptors, sodium ions and the potassium ions. In the last lecture, in the external fertilization, what the key steps आपण बघितलेले होते. There are three. These are the key steps. First one is what the chemo attraction is there. Chemo attraction मध्ये काय होता है कि there is a calcium ion influx is there, and there is sodium ions also influx is there, and there is a change in what the membrane potential takes place. Okay, and then afterwards काय आहे, एकदा का स्पर्म जाऊन बाइंड कुठे झालेला आहे, कुठे गेला आहे, sperm contacts to the jelly layer, then it contacts to the acrosomal reaction, and that after the acrosomal reaction, then binding is going to bind on the EBR receptors one. These are the EBR receptor 1, and then sodium ions is there. आणि मग आपल्याला काय बघायचं आहे into this. These are the four graphs, 1, 2, 3rd, and the 4th one is this. In this, which is in which and this is the graph for the unfertilized egg is there, but in that which is for the fertilized egg is correct. From this graph, the expression of EBR1, expression of the sodium ions, and expression of the potassium ion by its concentration, there is a shows that which egg is being fertilized or not is there. We are going to study this first graph, second, third, and fourth, in which all of you can also think over it, which is the graph is correct for the fertilized egg. In unfertilized egg, it is thought to be like this way. The EBR receptor 1, that is which is shown into the white colored box is there. Then sodium ion, it is a black in color, and the potassium one. This is for unfertilized egg. And what happens to the fertilized egg? By which graph it is shown that the given egg is being fertilized? How it can be identified that? Yes, anybody knows? Yes. Yes, Varsha.
Yes, ma'am.
हा. Do you know in this four graph which graph shown that the given egg can be a fertilized egg? What happens to the when if egg is being fertilized? What are the different events takes place into the egg receptors due sodium ion concentration and the potassium ion concentration? काय होत आहे? What happens? Which graph shows there is a fertilized egg is there? Yes, Nikita. Varsha, can you tell me?
मॅम रिपीट करा ना अजून एकदा.
Okay. This is the graph for the unfertilized egg and unfertilized egg. This is the graph for the unfertilized egg, and these are the four options given to all of you. The first, second, third, and fourth graph is there, by which all of you have to find the which graph is correct for the fertilized egg is there. Which graph is for fertilized egg is there? What happens in a fertilized egg? What regarding to the EBR receptor 1, sodium ion and the potassium ion, by which we can identify easily. Now, no, this graph is for the fertilizing. This graph is for fertilizing. आता in general आहे, मी तुम्हाला सांगते what happened into the fertilized egg with regarding to the EBR receptor 1 and sodium ions. Varsha, look, listen carefully and tell me one by one. Okay, what happened? First of all, the chemo attraction takes place into that reject going to secreted by egg, and which is joined to the sperm head on the RGC receptors, and then the sperm is moving from the aquatic environment towards the egg is there. Okay, after 1/8, there is a sperm is going to bind on the jelly coat. This is the second step. And then second step नंतर काय, third step is what? It is going to acrosome reaction takes place. It will form hole to the jelly coat, and it will bind onto the vitelline membrane onto the EBR receptor for the sea urchins. And if it is a mammal, then it is a ZP2 receptors. In yesterday lecture, we discuss about this. By said one receptor of the sperm is there. ठीक आहे. But EBR receptor वरती जाऊन bind होते onto the vitelline membrane is there. There are two events happen to the egg. What happened? One is what? Sodium concentration is going to increase to one. We discuss this, because of which what the fast block it is called it is seen in the external fertilization is there. External fertilization मध्ये तो बघितला गेलेला आहे. Into the internal fertilization it was totally absent is there. Okay, it only the slow block is present, which is a permanent block is there. म्हणजेच काय आहे, fertilized egg सपोज आपल्याला बघायचं आहे, we have to study it. What happens to it? To EBR receptor ones are काय आहेत, सगळे काय झालेले असतील, egg वरती प्रेझेंट असणार आहेत. That means their concentration is increased one. In all the graph, EBR receptor 1 is going to increase. This white colored box indicates this is the EBR receptor 1. Okay, EBR receptor 1 काय झालेलं आहे बघा, increase झालेलं आहे in first graph also, in second graph also, in third graph also, and the fourth graph also. But immediate when the sperm is going to bind onto the egg is there to block the polyspermy, there is a fast block. मी तुम्हाला शिकवताना पण सांगितलेलं होतं की काय आहे, there are two concepts. One is a fast block and another is a slow block. Into the fast block, what happens? Sodium ion concentration get increases, and so membrane potential is going increasing from minus 70 millivolt to the positive plus 20 millivolt is there. And because of that, it is the because of changing the membrane potential, there is no other sperm is going to bind on the that egg one, and which blocks the polyspermy basically, and which is a fast block one. It is regarding with the fast block is there. म्हणजेच काय आहे, sodium ion concentration is also increases one, and there is no role of the potassium ion discussed in this one. म्हणजे सोडियम आयन कॉन्सन्ट्रेशन, पोटॅशियम आयन कॉन्सन्ट्रेशन वाढते किंवा कमी होते हे डिस्कसच केलेलं नाही आपण, बरोबर आहे. म्हणजेच काय बघायचं आपल्याला, EBR रिसेप्टर 1 कॉन्सन्ट्रेशन वाढलेलं पाहिजे, सोडियम कॉन्सन्ट्रेशन पण काय झालेलं पाहिजे आपल्याला, इन्क्रीज झालेलं पाहिजे आहे. आणि पोटॅशियमचं काय आहे, देर इज अ नो काय विथ द पोटॅशियम आयन, ते इन्क्रीज हो नाही तर कमी हो, देर इज अ नो रेफरन्स विथ द these potassium ions. इथे बघू शकता, in the first graph what happens, EBR receptor 1 is increased but sodium concentration is not increased one, it is increasing but little but one, no, it is increased in very higher in amount. and in K plus ions पण काय दाखवले इथे, very higher concentration increase झाले. तर हे कुठेच डिस्कस केले गेलेले नाही. दॅट मीन्स फर्स्ट ग्राफ डू नॉट शो दॅट, म्हणजे फर्स्ट इज नॉट करेक्ट ग्राफ. सेकंड ग्राफ मध्ये पण त्यांनी सेम दाखवलेलं आहे, देर इज अन इन्क्रीज पोटॅशियम आयन्स, म्हणजे हा सुद्धा ग्राफ नाही आहे. फोर्थ पण ग्राफ बघू शकता, त्याच्यामध्ये काय दाखवले, सगळीकडे EBR रिसेप्टर तर आलेच पाहिजे आहेत ऑन टू द व्हायटलिन एनव्हलप. पण इन लास्ट ग्राफ मध्ये तुम्ही बघू शकता आहे की EBR रिसेप्टर 1 एकदमच कमी झाले. इट कॅनॉट बी हॅपन टू द फर्टिलाइज्ड एग, किंवा फर्टिलाइज्ड एग साठी काय आहे, EBR रिसेप्टर शुड बी लार्जर इन अमाउंट ऑन टू देअर सरफेस, म्हणजेच काय आहे की त्याचं इन्क्रीजिंग अमाऊंट पाहिजे. द थर्ड ग्राफ, तुम्ही थर्ड ग्राफ मध्ये बघू शकता की इन दॅट देर इज increasing in EBR receptor 1 and there is increasing in the sodium ion concentration, but potassium ion concentration should be same in the unfertilized and the fertilized egg. That means the third graph is being correct. Like this way all of you have to solve the questions into the MCQs one. Okay, this is the graph for the reference and this is graph for the options is there, first, second, third and fourth. We have to choose correct graph for the fertilized egg. ओके, फर्टिलाइज्ड एग म्हणजेच काय होतंय, फर्टिलाइज्ड एग मध्ये काय होतं आहे, देर इज अन इन्क्रीजिंग अमाउंट ऑफ EBR रिसेप्टर 1 सोडियम आयन. पोटॅशियमचं काही सांगितलेलंच नाही, सो वी कॅनॉट डिस्कस ऑन इट. म्हणजेच काय आहे, फर्टिलाइज्ड आणि अनफर्टिलाइज्ड एग मध्ये त्यांचं कॉन्सन्ट्रेशन कसं असणार आहे, सेमच असणार आहे. लाईक दिस वे यू हॅव टू थिंक, यू हॅव टू डिस्कस इट. ओके, वर्षा कळालं आहे?
हो मॅम,
आर
कळालं.
यू गेटिंग मी? नेहा,
येस मॅम.
both the Nehas, Yogita,
Yes, ma'am.
ready? Okay. This this is the question regarding to the external fertilization is there, and this is very common question, can in every exam it can be asked. Okay, but the option can be changed, question pattern can be changed, but common is these graphs are the common one. Okay, remember in mind that all of you. This is the structure of the mammalian eggs. Mammalian egg मध्ये आपण बघितलं होतं, there is outer membrane which is a cumulus, corona radiata cells are there, perivitellin space is there, zona pellucida is there, then into that there is a nucleus, and into that cytoplasm cortical granules are and the mitosis are there suspended into it. Then these are the key steps into the internal fertilization: chemoattraction, the capacitation, acrosomal reaction, binding of sperm to the extracellular coat is there, binding to the zona pellucida and ZP2 receptor. There is one ZP2 receptor is important. Why there is and it is a decent discovery basically. In last lecture we discussed about this, okay, how the ZP2 receptors are important into the mammals one, and which gives the specific binding. In that we discussed in yesterday lecture about the mouse experiment. In that we take the 4 transgenic oocytes, and with that we added 4 transgenic oocytes which expresses human as well as its mouse. The receptors are there, ZP1, ZP2, ZP3, which are coming from the mouse, and in the humans the ZP1, ZP2, ZP3 and ZP4 receptors are present, and all the receptors are presented by these mouse eggs are there, in which solution we added the sperms into it, but the human sperms are going to bind only on the human ZP2 receptors only. They cannot bind on to the ZP2 receptors of the mouse is there, and it can be shown that there are 2 meanings in it. म्हणजेच काय आहे की it is a specific reaction is there, and onto the ZP2 receptor there is a binding of sperm takes place. Okay, this is the diagram for that experiment is there. Then we discuss about the fast block and the slow block into the mammals is there. Fast block is absent and slow block is given by the calcium ions, which activates. Now the calcium ion concentration is increasing into the cytoplasm by the phospholipase C zeta, which is coming from the sperm head and which is translocated into the cytoplasm, which act on the PIP2, which gives the secondary messengers IP3 and the DAG. Okay, then IP3 is what? The inositol triphosphate, which then activated the SER, smooth endoplasmic reticulum, and which activates the calcium channel activation, which activates the calcium channel, and then there is a formation of the multiple way of the calcium ions, then there which is released into the cytosol, binds to the calmodulin protein, captain kinases activation, protein kinases activation, which eliminates the translocator inhibitors on the mRNA, and by which the protein synthesis can be start. But side by side, this is important point in the last lectures internal fertilization मध्ये काय बघितलं होतं आपण की there is a degradation of the moss, that is a cytostatic factor, and because of which the arrested egg which is arrested in the metaphase 2, now it will continue into the completion of their oogenesis. It will form the mature ovum by the formation of the anaphase promoting factors and by degradation of these moss cytostatic factors, and there is a then there is a formation of the ovum and the secondary polar bodies, which are then degraded because there is a no use of that secondary polar bodies is there, and then afterwards there is a fusion of female nucleus pronucleus to the male pronucleus takes place, and then which causes side by side मी तुम्हाला आता सांगितलं there is a simultaneous processes are there in which there is a complete block to the polyspermy is there by the removal exocytosis of the cortical granules, and there is a release of the ovastatin, and there is a removal of the ZP2 receptor is there, and then there is a fusion of male and female pronuclei which activates basically what DNA is very tightly packed of the sperm is there, and which is being uncoiled by the enzymes protamines and the glutathione is there, and then there is arrangement of these chromosomes onto the metaphasic plate. There is a degradation of the nuclear membrane takes place. There is a chromosome alignment at the metaphasic plate, and there is a then cell division starts. That is a next phase of the development, that is a cleavage is going to start. Okay, any doubt regarding this? Yes, anybody? If all of you have any kind of doubt, all of you can ask me. By this way, we completed our the internal fertilization and external fertilization revision also. If you want any kind of doubt, all of you can ask me. Okay.